Countdown begins. So today we're going to be going over genetic material, also known as DNA. So today, before I get jumping into our little lab demo, I'm going to go over a couple of key concepts before we get into this. So first of all, I want to ask everyone, basically, what do you guys know about DNA already? And I want you guys to talk amongst yourselves. So for this purpose here, though, we're going to speed through that. That would be my introduction. Then through there, now I'm going to go through the different topics. So I'm going to start off with DNA. So this actually stands for a really long word, which I'm not going to write on the entire board, but deoxyribonucleic acid. It's a specific molecule, and it is basically the inherited material responsible for variation. And we have a nice little drawing of this as well, which I will explain as well in a little bit. Basically, DNA is packaged into chromosomes inside of cells. Think of DNA as basically a really long strand. For instance, a typical human DNA strand is about like 2 meters long, so they have to be condensed down to be able to fit actually inside the cell. So they're then put into chromosomes, and then through there we have genes, which are segments of the actual DNA, which correspond to a single uninterrupted segment, which contains coded instructions for the cell, which is really cool. So, before I get to the demo, one more question, because I know everyone likes questions. How is DNA used in the real world? So, some classic examples, basically like little Jimmy over there just said, used in like, basically in crime scenes to determine who the culprit is. In laboratories too, let's say for instance, the effects of diseases, they use DNA to see the effects. So, now we're going to get straight into the little demo I have for you guys. You should be taking notes while I'm doing this. So we're going to start off with our lovely little strawberry. We're not going to eat them. It's going to go straight into the bag. I made a special solution containing soap, salt, alcohol, and also water, which we're going to be adding into here. Poured out 20 milliliters of it already, so that's going to go into the bag. And we want to make sure we get out as much air as possible from our Ziploc bag when we go to do this. Now, can anyone think of, for instance, why I ended up putting soap, salt, and alcohol into the solution? So, since little Timmy over there is really into this, gave us a couple of little examples. Not quite exactly what we wanted, but this is kind of the type of answer I'm looking for. So to help clump up DNA together would be the purpose of the soap. Then the alcohol to dissolve everything except for the DNA. And then for the salt, oh, actually the salt was to clump up everything together. My bad. Misread that, my handwriting. Alright, so with this, I have to crumple this guy up for a couple of minutes. So I got some more questions for everyone before we continue on. So where is DNA found in the cell, for instance? Oh, Fiona, good one. It's found in the nucleus. That's one there. And just so happens that all living organisms contain DNA. We're going to go into... So, what does DNA look like? As you've seen, I've drawn up here on the board very simple structure of DNA here. So we have the double helix backbone, then we also have the different pairings of specific molecules: thymine, cytosine, adenine, and guanine. These guys here are important in encoding genetic information for the cell to use. So, now that this guy is pretty much mushed up, I'm going to go ahead, got this, and start pouring this out into a test tube and filtering it. We're going to use our basic technique here of Folding our filter paper so that it fits. Nothing too fancy. And then while we're waiting for that to drain, I got some more questions for everyone. I just love questions, and this thing does not want to stay in. And this is why we're going to end up doing this in pairs later, so that you have a helping hand when you go to pour all your material in here. Also help it along a little. Just make sure not to rip your filter paper. Would not be good. Got pretty much enough in there for now. Let that settle for a little bit. Put that in the bigger test tube. That was my mistake. There we go. So I have enough room for the next step, which is the rubbing alcohol, which will help separate the DNA from the material we just crushed up. Ooh, is there enough room? Yes, there is. Now, while we let that settle a bit, so what does DNA look like? Any ideas out there? Well, it's going to come up stringy and white. You guys will see it in a little bit. It'll eventually end up coagulating into the alcohol there. You can actually see a little bit of it already. So going back again to the double helix structure, like a twisty ladder. And from there, DNA. So what are some traits that are DNA that helps make us unique? So like, for example, eye color, height. Those are some classic examples. Do all living things contain DNA? All living things. Actually, even dead things contain DNA. For example, mummies and things like that. They're able to extract DNA out of those old corpses and able to assess the ancestors of the mummy. Here's a couple of other key questions. The purpose of reproduction. So, scientifically speaking, to pass on characteristics from parents to offspring, which is kind of cool. And we also have what is variation? So diversity in a species. So for instance, everyone in the classroom, they all look different, but we're all part of the same species, Homo sapiens sapien, which is kind of neat. And then what causes variation? So it's the exchange of genetic material within a population. So with that, I'm going to go ahead, and this should be about done settled. So I'm going to go in here, about an inch in, just stir it up a little bit, and then I should be able to just pluck out some of the DNA. I think I made it. Oh, there it is. And get in there. And it worked. So, now you guys have all seen how to extract DNA from strawberries. I'm going to have everyone go ahead and do the lab and make sure you're wearing your gloves, your eye protection as well. And there's also the eye wash station. If you get rubbing alcohol in your eye, we're going to have to wash it out right away for 15 minutes straight. So don't get that in your eye. And at the end of your actual lab, you're going to hand in your exit slip, which has an answer, which has a question that I want at the end answered. And then if we have time, we'll go over different genetic technologies. So for instance, there's some cool examples of Dolly the sheep and fluorescent rabbits that I'd like to discuss with you about. So that's about it.
I think I paused it.
Oh, didn't